What we're going to talk about now is how we produce force with these twitches. So what I've drawn here, which you can see in front of you, is one sarcomere with one thick filament and the surrounding thin filaments, but in three different stages of contraction, or three different lengths. So you can see at the top, it's the shortest length of the sarcomere. You can see that the Z discs, those backbones, are right up against the ends of the thick filament. In the middle one, it's at sort of a medium length, and in the bottom one, it's at a more extended length. And over here on the side, we're going to have a graph of sarcomere length versus force generated in a twitch. So if we start with the sarcomere at one particular length, how much force can we build up in the twitch? And that's going to be based somewhat on how many myosin heads can reach the actin. So let's start with, say, this shortest sarcomere, the one on the top. And imagine that we deliver one action potential to the muscle cell that this sarcomere is in, and see how much force it can generate. Now you can see that all of the visible myosin heads can reach the actin. So all of them can grab on and pull. So that's going to mean a lot of force from there. But notice that it doesn't have very far to go. Those thick filaments are going to run into the end of the thin filaments, which means it can't actually generate very much force. It can't build any force during the twitch. So with a short sarcomere, the amount of force generated will be relatively low. We'll put something here. Now take a look at the middle one. You'll see that in that one, pretty much looks like all of the myosin heads can reach an actin. So here I'm talking about that this myosin head can reach an actin, and so can this one, and so on. All of the myosin heads are where they can reach an actin. So when the tropomyosin gets out of the way, all of those myosin heads are going to be able to contribute to the force of the twitch. But it also has some room to go, so they'll have the ability to do several contractile cycles, building up tension, which means that for a sort of a medium-sized sarcomere, the amount of force generated will be higher. Now look at the bottom one. In this one, while there's lots of room for contraction to happen, many of the myosin heads, when you move the tropomyosin out of the way, don't have anything to grab onto. So they can't actually do their contractile cycle. They can't grab an actin, meaning they're useless. They can't contribute to the force of the contraction. So as the sarcomere gets longer, after a certain point when those myosin heads start coming out from under the thin filaments, the force drops off again. And if you were to graph this, what you'd have is a graph that looks something like this, where there's an optimum length somewhere in the middle. And then if the sarcomere gets longer than that or shorter than that, it loses some of its ability to generate force. One way to think about that is this. It's been a while since I've been able to do a pull-up, but my understanding, if I remember, is that if you start from here, you can do it more easily than if you start with your arm fully extended. And a lot of times people think that's because if you're here, that means the muscle has already started contracting. But it's kind of funny. If you stand on a box so that when you're gripping the bar, your arms are bent, so there's no force there, it's still easier. And the main reason is if you think about the sarcomeres here in your biceps muscle that's going to be contracting this joint, when it's fully extended like this, they're stretched out. So some of the myosin can't reach the actin, which means when that muscle contracts, right at first until it contracts a little further, it's not going to be very strong. Likewise, if it's already here, there's a limit to how much more it can do. It can't generate that much more force. But there is some medium point where it's at its strongest. And that's what we're looking at here with sarcomere length. So this is what's sometimes known as the length-tension relationship. The interaction between the length of the sarcomere in a muscle and the tension that you can generate in a muscle twitch. Alrighty, so now that we know that, that's actually just something of a point of interest. What our next point is going to be to talk about is how do we go from twitches to generation of force over time in a muscle? Because when I tell this muscle to contract, it doesn't just go like that. Instead, I can cause it to contract and then sustain force. So how do I do that if one action potential produces one twitch? Well, let's see. So we will get rid of those. And we're going to put up a large graph here. It's going to be a graph of tension versus time. And on this graph, an action potential will be represented by one green line. So that's saying at that point in time, that muscle had an action potential. And if we look at the force generated, we will see a muscle twitch. One action potential produces contraction followed by relaxation. So what happens if we deliver another action potential? Well, we get pretty much the same twitch. One action potential, one twitch. So we might ask, okay, what happens if I deliver two action potentials where the second one comes before the first twitch is done? And in that case, here's what I get. The second twitch adds on to the first one. I build up more tension in that muscle fiber. I should make a note. Here we are talking about tension in a single muscle fiber. This is important to realize. This graph is about what's going on in one muscle fiber, not the muscle as a whole. That's going to be a little different. So here what I see is that if I deliver a second action potential, I get a second twitch, but it kind of adds on to the first one. If you think about what's going on down at the molecular scale, the first action potential went down the T-tubules, hit DHP, opened the ranitidine, calcium came out, got into the myosin, got into the myofibrils, bound troponin, troponin, tropomyosin, and we started doing contractile cycles. The calcium pumps are working, so they're taking the calcium away. So we get through maybe 3 contractile cycles, and now the tropomyosin is blocking us again, so we start to relax. And then the second action potential hits, and we send out more calcium. So imagine this is my thin filament. Here's our starting position. We did 1, 2, 3, and then the tropomyosin blocked us, so we started to pull back, and then the next wave of calcium hit. We got our second twitch, and notice we don't start from where we started. We start here. 1, 2, 3. We were able to build up more force because we didn't go back to the starting position. So our second twitch added on to the first. We call this idea summation. Summation is what happens when one twitch adds on to another. When it happens before the first twitch is done, so it adds its force to it. One twitch adding on to another. All right, so we might wonder, well, can I do 3? And we find 1, 2, 3. As it turns out, we can. So then we might ask, all right, how far can I push this? What happens if I just keep having action potentials? And what we get is this. You'll note that in this example, we are now sustaining force in that muscle fiber over time. If we were to look at that fiber, it's going it's holding that force. It's exerting force over time. Sustained force over time. So sustained force over time is what we call tetanus, or a tetanic contraction. We have put this fiber into tetanus in this example. Specifically, this is what we call an unfused tetanus. It's tetanus because we're sustaining that force over time. It's unfused because the twitches still show a little bit of difference. There's a tiny little relaxation, slight relaxation between each twitch, because the action potentials are spaced out far enough that the calcium pumps get to let the tropomyosin block for a moment until we remove it again. Which means then we might wonder, okay, well, what if we don't give it time? What if I deliver action potentials very quickly? So that in this case, think about what's happening. The action potentials are coming in so fast that the DHP receptors are more or less constantly stimulated, and the ryanodine receptors are being held open, which means calcium is just continuously flooding out. Now think about what happens with the sliding filaments. Now there's always calcium present. The calcium pumps can't keep up with it. So there's always calcium on the troponin. The tropomyosin is always out of the way. So our myosin is able to just do continuous contractile cycles. What should it look like on this graph? Will there be any relaxation periods between the twitches? My guess would be no. And what we'll see will look like that, which we call fused tetanus. Fused tetanus is the muscle fiber developing the maximum possible tension. That's what happens when you're just holding the tropomyosin out of the way, and those myosin heads are working as fast as they can continuously, and the muscle contracts as far as it can, as hard as it can. That's a fused tetanus. Now, this is all in a single muscle fiber. So if I want to take a single muscle fiber and get it to produce force over time, I need to have twitches fast enough that they summate into a tetanic contraction. Summation is what we call it when twitches add onto each other. Tetanus is what we call enough summation to hold the force over time. Unfused tetanus is when there's a little bit of relaxation, sort of a bounce. Fused is when it's just solid. So unfused is like that. Fused is like that. Make hopefully a little sense. Now, this is not how we normally produce force in a muscle as a whole, for a couple of reasons. Number one, this is exhausting to muscle fibers. Any kind of tetanus where they are forced to stay contracting over time is very tiring. Many muscle fibers can only do that for a short period of time before they stop responding. The other reason is that something like fused tetanus is actually dangerous. As we mentioned earlier, muscle fibers are stronger than you think. And if you drive a lot of the muscle fibers in a muscle into fused tetanus, that muscle may contract hard enough to hurt you. So, in a very strong sustained contraction in a normal muscle, you might see some of the fibers going into unfused tetanus. Fused tetanus is not something you can normally do. That's what you get with really strange ionic imbalances, or grabbing an electric wire or something like that, where the electrical shock is just causing your muscle to contract out of control. So unfused tetanus you will sometimes see in muscle fibers during a very strong contraction. Fused tetanus you do not normally see. Okay, so that's what's going on in one muscle fiber. So how do we produce muscle contraction in the muscle as a whole? That's a slightly different question. Actually, it's a very different question. So in the muscle as a whole, let's see, I have a decent Excel spreadsheet that I can use to show this, and I'll bring that up too. Maybe I'll walk through that in a supplement here. But what I'm going to do here is have you imagine that we have multiple muscle fibers. Actually, it would be multiple motor units, but we'll represent each motor unit as one thing on this graph. So you could imagine that we have, say, the red motor unit. And we're going to stimulate it so it twitches, 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 like so. So that's our red motor unit. And we will have an orange motor unit. It's going to do the same thing, but slightly out of sync. Now notice, these muscle fibers or motor units are not going into tetanus. These are separate twitches. And let's see, here will be the green motor unit. And the blue motor unit. And the purple motor unit. Now up here at the top, we'll put in what you would get if you added those together. So if we look at this moment in time, all we get is the force from the red motor unit. And here we get what's going on in the red plus what's going on in the orange. And here, the red is dying off, but the orange and the green are both involved. Here, the red is gone, the orange is dropping off, the green knows maximum, and the blue is kicking in. And what we would get would be something sort of like this. In other words, sustained force over time. Because what we have here is each individual motor unit twitching, and then while it's relaxing, other motor units are twitching to take over the load. So they're sort of sharing it. It's sort of like saying, okay, I'll pull, okay, I'm tired, okay, I'll grab it, okay, and now I'll get it, now I'll get it, and so on. So there's always somebody pulling, which means that each person's going, okay, like that. You're generating a continuous force. This is how our muscles normally do it. If I'm going to hold up this pen, when I lift it, no fiber in here is undergoing tetanus. Instead, they're going twitch, 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 all over the muscle, so that at all times, some group of motor units is contributing to that contraction. That way, I get a continuous steady force by everyone passing it around amongst themselves. The name for that process is asynchronous recruitment. The process by which we have multiple motor units asynchronously, out of sync with each other, take turns twitching, so that there's always somebody involved, usually always several people involved, usually, which produces sustained force over time. Now looking at this graph, how could I get more force? I can see two ways. One, bring in more motor units. So maybe at the same time red is going, we've also got pink. And at the same time orange is going, we've also got chartreuse. And just whatever you want to call it. But more motor units adding to this would generate more force. Another way would be to get red to go more often. So rather than being spaced out like they are, bring those closer together. So still, at any given time, more people would be doing it because each of them has a faster cycle. So both of those are ways that our muscles can generate more force. If I want to hold up a 5-pound weight, that's not that much. Not that many motor units have to be involved, and they can rest between. 10-pound weight, more of them have to be involved. 30-pound weight, probably not just most of them involved, but they're twitching pretty fast. 50-pound weight, they're pretty much all in. Now, in the next lecture, we're going to talk about how muscles use energy and how not all motor units are the same. There's different kinds of muscle fibers, and we recruit them. We bring them in a specific order to maximize efficiency. Some of them are stronger than others, and we leave those till later only if we need them. But this idea of asynchronous recruitment is that using whatever ones we've got, we rotate among them. Now, if you haven't done the EMG lab yet, you can keep this in mind when you look at what an EMG looks like. And if you have done the EMG lab, this may inform you on that. Think about what this means. What would I expect to see electrically in this muscle? So let's see, there must have been an action potential here, and another one here for the orange, and here for the green, and here for the blue, here for the purple, and there's another one for the... So electrically in the muscle, I'm going to see lots of action potential. And we would obviously have many more than 5 motor units. So when this muscle is active, what I expect to see will look like this. Lots and lots of individual action potentials, each of which is short in duration, millisecond or two. But there's a lot of them kind of happening rapidly in rapid succession. What that's probably going to look like, if I'm looking at sort of the whole muscle like I do in an EMG, is static, noise, but more noise when more motor units are participating. Louder noise, if you want to think of it that way. So that will that will inform either what you have seen if you've looked at the lab, or what you will see when you look at the lab. Okay, in our next lecture here, we're going to be talking about muscle energy, how muscles get their ATP, and then a little bit about the different types of motor units. That does mean we're going to be going over glucose metabolism, which you may have done in an intro bio course. So leave plenty of space in your notes for that one. Okay, I'll see you then. Here's another way of looking at the idea of asynchronous recruitment. So the idea here's what it might look like if I had a single motor unit contracting. Don't pay any attention to the numbers on the X or the Y axis. They're just numbers. But here, blue motor unit is contracting, and you see that they are separate twitches, so we're just getting an amount of force each time. Now imagine that I had another motor unit that was also twitching, but a little bit out of sync. If I add together the force that those two motor units produce, this is what it comes out as. Now imagine adding a third and adding that into their added together force. And then a fourth. So you can see that as I'm adding in additional motor units, it's not just that the force I'm getting is larger, but that it's now lasting longer. Add in another one, and another one, another one, and finally another. And now what you can see is that the force generated by all of these motor units is now being sustained over time. This is the idea of asynchronous recruitment, that if each of those colored things on the bottom of the graph is a separate motor unit within a muscle, then by having many motor units twitching slightly out of sync, the total force generated by the muscle is now sustained over time. So each individual motor unit can rest a little bit.